Yes, thank you, Mateo. And it's my pleasure to give concluding remarks for this amazing two-day session looking at modeling. But before that, I want to actually announce the poster prizes for our session. So that I have time to say everything I want to say. So the poster prizes we decided based on the quality of the posters, the abstract, and there was equal weight or little higher weight was given to the reviewers who reviewed the posters. And then we also took into account what the community had to say through the voting that we announced yesterday. So it was the poster assessments were performed using these multiple different criteria, mainly looking at the scientific content, presentation, and discussion at the poster session. So with that, our first prize, we had to split between two amazing posters. The two posters are automated whole cell modeling from genomic sequence and multi-omics data. And the second one that got the first prize is an agent-based model of tumor-associated macrophage differentiation in chronic lymphocytic leukemia. The second prize goes to Boolean network inference at different levels of logical complexity. And the third prize goes to simulating drug effects on whole cell level simulation. So congratulations to all the students and the postdocs who presented these posters. Thank you for your contribution. And I think there is another announcement at the whole ISMB level. So with that, I'm going to share my screen and give some concluding remarks. You see my screen, Mateo? Okay, I will expand it. Okay. So it's my really pleasure to conclude this two-day session, the SysMod meeting. I think SysMod is a very interesting group of scientists that loves to discuss multiple different avenues of computational biology. And I think our community is really interested in integrating mechanistic models with large-scale data or even small-scale data and really trying to validate some of the outcomes of modeling. And then, of course, as more and more omics data is becoming available, we are integrating, as we saw in multiple different talks, we are integrating approaches to model with bioinformatic analysis and the statistical analysis to develop higher level integrative models, and still trying to think about the local effects and validate these local responses in different systems. And some of the systems we heard today are looking at bacterial metabolism, human data, plants, and yeast. So this is really integration of these multiple different approaches to understand what are the mechanisms for understanding biological systems. Some of the modeling techniques that I would like to highlight, and I do still highlight more in the later part of my talk, are dynamical modeling, flux balance analysis, logical modeling, network modeling, and stochastic simulation. So these are some of the approaches that we saw in last couple of days. And if there are any questions that that community has today throughout my talk, please just interrupt and ask, or Mateo can ask for you. I think it's really, this is discussion, and we would love to hear from you. What are your thoughts from this two-day meeting? So on the first day, we started with a keynote speaker by Ines Thiel. She spoke about whole body metabolic modeling, providing novel insights into host microbiome crosstalk. I really, so some of the things that she talked about was, of course, developing metabolic models that can integrate knowledge base, mechanistic models, computer models with the high quality genome interactions. And I think she really emphasized these, the inclusion of high quality genome interactions in our in these mechanistic model, and using those to really make predictions that are useful and can be validated experimentally. I really like this analogy of explaining how the analogy that she used to explain how these models work, and we are trying to understand the flow through the system, and then there are regulators that can regulate these flows and the signaling flow. And we are developing most of the talks that we heard are trying to develop these techniques to understand this signal flow and use that in a useful manner. So then she spoke about virtual metabolic network that has been developing for last 15 years. And then Demeter pipeline that includes graph construction, reconstructed microbiome, and a network of microbiome interactions. I really found it interesting how the multiple different ways that she used to discuss the whole human microbiome interactions. So she talked about whole body model that includes organ-specific metabolic maps, including 28 organs and 13 biofluid compartments. There were 57, she modeled 57 inherited metabolic diseases using this network, and then added microbiome to understand the consequence of different microbiome compositions. She also took into account drug microbiome interactions, thinking about around 5,000 strains, 15 drug metabolizing enzymes that are encoded by 25 microbial genes. And so then she's focused on colorectal. She gave specific examples of colorectal cancer patients, and looking at human microbiome interactions in these patients, then also thinking about brain neurotransmitter production and how microbiome can regulate that. And then really trying to apply this to the personalized level so that we can use it to create personalized treatments for patients of these different diseases. The keynote speaker talk was was followed by a session on disease and multi-scale modeling. Sorry about that. Okay, disease and multi-scale modeling. So in this part, we covered different modeling approaches to address questions regarding a variety of pathological conditions such as cancer, psoriasis, type 2 diabetes, rheumatoid arthritis, multiple sclerosis, and primary biliary cholangitis. I think I'm going to have to move a little bit faster. So there were discussions on the nasal community model, combining two different pathogens. We spoke about, we heard about a toolkit for concretely describing non-canonical polymers. And then there were two tumor models. One was yesterday actually, and one was today, that these two models were trying to understand how tumor proliferates in extracellular matrix, and the modeling approaches that they used were PZ-cell and BOS, which combines the Boolean modeling of signaling pathway along with the spatial spatial modeling, and also phase field model that we just heard today. Then there was logical model of psoriatic keratinocyte and glioblastoma multiform. We heard about glucose insulin model in type 2 diabetes using differential equations. This model had 36 parameters which were estimated from non-diabetic and diabetic individuals. We also heard about metabolic model of CD4 cells to study drug repurposing in different autoimmune diseases. And this model tried to develop constraint-based model to understand how CD4 cells, focusing on naive T cells, CD4 cells to Th1, Th2, and Th17, and how these cells respond to different drugs. So then we focused on infectious disease modeling and heard about ordinary differential equation modeling of phosphorylation of STAT and altering the ratio of diffusion constants of interferon and virus in the cell culture and how it affects the plaque growth arrest. We heard about this algorithm which uses single cell RNA-seq data to understand dysregulated signaling in people who are living with HIV and what are the drivers of a higher prevalence of atherosclerosis in these HIV positive people. Then we heard about two genome scale metabolic models to characterize mucin-driven shifts in bacterial metabolism and to model condition-dependent production of phenenzymes. So then there was today's talk, and I'm going to do a brief overview of today's presentation since they just happened. So I think Ruth Baker gave a very nice presentation describing very simple models, but yet extremely useful. And she used these Bayesian and ODE models to study regulation of RNA protein complexes and how it controls mRNA expression in Drosophila. Then we heard about the stochastic cell motility model and how the cells move in response to preferential polarizing. And she also again used synthetic based for parameter estimation. Again, that was followed by several exciting talks describing implementation of Enpadro in Python, then the classifier describing mRNA-based logic for grouping the cells. We also heard about rejection-based Gillespie model, which can model the presence of non-Markovian processes. These are a couple of talks which just happened, so I can actually skip that. So we just heard Dr. Holodinko talk about his rule and structure-based model of RAS to ERK's cascade and application to drug treatment. I think highlighted modular response analysis and how it can express the global response in terms of local analysis, and it can also address the network reconstruction problem, how network adaptations can improve signaling after drug treatment, and kinase dimerization can lead to drug resistance. There were 30 excellent posters we received, and describing various approaches of mechanistic models, including agent-based, constraint-based, and logic models, stochastic and deterministic approaches, and ways to improve knowledge base. So I want to highlight some of the different modeling techniques that I have been highlighting throughout this talk. But I think most of us were including logic-based, ODE-based, and constraint-based modeling approaches. There were a lot of improvements to existing methods and a lot of integration with omics data and different databases that are available in the public space to improve models and improve predictions and also improve optimization and the speed of the algorithms. Some of the caveats or the questions going forward in my mind, and I think that I also heard in our roundtable discussion, are how to make these mechanistic models available in the world when there is more and more omics data is coming. on our plate, and how we can actually describe these mechanistic models efficiently. How can we, one of the question and important question is, we need standardization for these molecular networks and all the models, and people are investing more and more into this problem, and that I think is improving mechanistic modeling field in general. But some of the questions are, can we use text mining to expand our the models that are available, and really use additional information to improve the structure of existing pathways and databases. I think my time is almost up. So, and then one of the difficulty in application of mechanistic models for human data is that there is no time course or the perturbation data. It's frequently not available. So some of the things that are modeled in the smaller organisms, such as yeast and bacteria, are not easily cannot be translated easily to the human, though we also heard about some ways to do that, which is, I think, very exciting. And another last point that I want to raise is, can we use machine learning techniques to improve some of the parameter estimation for these mechanistic models? So with that, thank you for everyone for attending this session. And that's all. If you have any last comments, I'm happy to hear about those.